All right. So, as usual, we had mentioned how, so far as scientists are concerned, everything is made up of what? Atoms. They interact by the process we call bonds, where we have like ionic bonds and what? Covalent bonds. Ionic is transfer of electrons, covalent is sharing of electrons. And then we have these bonds referred to as hydrogen bonds, where they um, are other bonds that are formed between molecules if they are polar. Because we said for covalent bonds we have non-polar and what? Polar. And for the non-polar, the electrons are being shared equally. For the polar, they are being shared unequally. And polar molecules can have what we call hydrogen bonds, right? And so all these interaction brings about the formation of what? Molecules. And with molecules, we have inorganic and organic molecules. And the main difference between these is the presence of what? Carbon and hydrogen for the organic molecules. And we said that these organic and inorganic molecules will come together to form what? The cell. And these organic molecules, we have four particular ones that are referred to as biological molecules because we see them in all life. Biological molecules. And they call them the building blocks of the cell as well. And then we said these biological molecules, we have four of them, which includes the carbohydrates, proteins, lipids, and nucleic acids. Okay, and they come together to give us a cell, which is made up of what? Organelles. Okay, and has a cell membrane. Okay. Has DNA or genetic material, and then we have organelles inside of what we call the cytoplasm. So all these details, we are going to talk about it later on. But basically, when we look at the cell, so far as scientists are concerned, we have two types of cells. We have the cells that humans have, and those cells we call them eukaryotic cells. And the cells that other organisms like bacteria have, and they call those cells prokaryotic cells. So today's topic is just on the eukaryotic cell, which humans are made of. Okay, we are going to focus on what? The eukaryotic cell. All right. Okay. So the human cell. All cells are made of these three basic components. All cells have a cell membrane we call the plasma membrane. They have genetic material. And cytoplasm. So these are the components of cells, whether they are eukaryotic cell or prokaryotic cell. Now, starting from the plasma membrane is made up of lipids and proteins, which may have carbohydrates added to it as modification. The genetic material is DNA and it's found in the nucleus. Okay, so we are looking at the human cell, which is made up of the cell called the eukaryotic cell. The cytoplasm has structures suspended inside of it, referred to as organelles. And then it has the fluidy component, referred to as cytosol. With the organelle part, there are some organelles that also have additional membranes around them, being inside the cytoplasm, so they call them membranous organelles. And then we have the non-membranous organelles. So we are going to look at the cell, and we are going to look at the structural components of it, which is basically these organelles, the plasma membranes, the nucleus and its contents. And we are going to look at their function, because anytime we do anatomy and physiology, we are doing what? Anatomy and physiology. Anatomy is what? Structure, and physiology is what? Function. So let's start with the plasma membrane. This is a cell. It has what? A plasma membrane. The cell contains what? A nucleus, right? And inside of the nucleus, we have what? A nucleolus. All right? Around the nucleus, we have all these membranous, flattened looking structures. And some of these, the ones around it, has ribosomes attached to what? Them. So they appear rough. And we have the ones that do not have any ribosomes attached to them, so they appear what? Smooth. And then we have these stack of looking like pancakes that referred to as the Golgi body. And then we have these other things that tend to contain substances in them. These are the membranous things. So we have the lysosomes and the peroxisomes. And then we have what they call the mitochondria. Of course, we have free what? Ribosomes all over as well. And then we have all kinds of cytoskeleton. All over. Okay. So first, we are going to start with plasma membrane. We said the plasma membrane is made up of what? Lipids and what? Proteins. So what's the smallest component of lipids? Triglycerides, good. Which is made up of glycerol, which has 3 carbons, and what? Each of them have what? Fatty acids attached to them. So a triglycerol is 1 glycerol and what? 3 fatty acids. So this is what happens. These lipids get modified by one of the fatty acids breaks off, and instead a phosphate is attached to the carbon. All right? So this part becomes hydrophilic. This part becomes what? Hydrophilic. Because of the presence of what? The phosphate. Because you know lipid and water doesn't mix. While these fatty acids, these 2 fatty acids left, becomes what? Hydrophobic. So they will call this the head, and then they'll call these 2 parts the tail. So if you look closely at a plasma membrane, the lipid part is modified because of the addition of the lipid. So it's called phospholipid, and it's 2 layers. So by what? Layer. So this is how it looks like. So this is the one what? Phospholipid. Now remember, lipids don't form polymers, so they are not forming bonds. They are just, it's just like how you are sitting next to each other. That doesn't mean you are Siamese twins attached. Okay, so the lipids are next to each other. Okay, in a layer, 1 layer and 2 what? Layers. So they call them bilayer. Are you with me? So it means one phospholipid can actually move and go to another position, depending on the membrane. So this is another. So we are looking at just a segment of it. Okay, and then this will, the tails face each other. So this is what? The phospholipid bilayer, which will be what? The lipid part of the plasma membrane. But the plasma membrane hasn't just got lipid, it also has what? Proteins. So the proteins may either span through the whole lipid bilayer, and they'll call those proteins integral proteins. Or they will attach to the surface either outside or inside of the phospholipid bilayer, depending on whether it's facing the cytoplasm or outside the extracellular part. So they'll call it what? Peripheral proteins. So the protein can be attached here, a protein can be attached here. Those would be what? Peripheral proteins, and then the protein can what? Completely what? Span the whole phospholipid bilayer, and that would be what? Integral. Does that make sense? All right. So basically, that's how the phospholipid bilayer, that's how it looks like. And sometimes we may have, let's say, a carbohydrate attached here and there, either to the lipid directly or to the protein to form glycolipid if it's attached to a lipid, or glycoprotein if it's attached to the protein. Okay, or just the lipid will surround the carbohydrate to surround it as a form of modification all around, like a glycocalyx. So it depends. If it's carbohydrate that the protein is attached, they'll call it proteoglycan. So it depends on it. So all these modifications can be seen. And all these protein they can serve as receptors to sense the environment. They can serve as channels. They can serve as cell recognition molecule. For instance, if you are your blood type is A, it means you have the glycoprotein A on your red blood cell. Does that make sense? So they play different roles. These proteins play different. Some of them can even act as enzymes. We'll look at all these things later on. And of course, some of them also act as anchoring proteins, because we all know we are multi-cell organisms. We are made up of a mountain of cells, but the cells are most of the cells are somewhat connected to each other. Imagine our cells are not connected. We just fall into pieces, pile of cells on the floor, just independent cells. So of course we have anchoring proteins that hold and glues and attaches adhere cells together. We look at these different kind of silver adhesion molecules as well. So basically, this is the plasma membrane structure. In terms of function, I've mentioned some of the functions of cell recognition as channels to allow selective permeability and stuff like that. And functionally, more importantly, one of the roles it makes is to bring about selective permeability to allow specific things to enter and leave the cell. Now, because of the plasma membrane, it has divided the contents of the inside of the cell is referred to as what? Intra what? Cellular. All right. And then the outside is what? Extra what? Cellular. It's one word. Okay. So because cells come together to form tissues, and so they'll cluster, like the in between cells in tissues, they'll call it interstitial. Okay, they'll call it interstitial, which is the same as what? Which is part of what? Extracellular. So anything blood is outside of the cell. Anything outside of the cell is what? Extracellular. So interstitial fluid, which is a fluid between cells, is also what? Extracellular fluid. While the cytoplasm will be, the cytosol will be what? Intracellular. So everything in the cell is called intracellular. All right. So that's the structure of the plasma membrane. Now, let's move on to the genetic material. The genetic material is what? DNA, right? So DNA is a polynucleotide. Remember we talked about that when we were doing the carbohydrate, proteins, lipids, and nucleic acid? It's a double-stranded what? Polynucleotide connected to each other by what? Hydrogen bonds between the bases, and is double helix, helical, anti-parallel, very long. And this DNA molecule is supposed to be located where? In our nucleus. It's so long that it has to be wound around something like a spool. So DNA is wrapped around. It's not just left like spaghetti swimming inside, or chicken noodle soup, noodles just what? Swimming in there. Is that the noodle has been wrapped around these proteins, right? These proteins are called histone proteins. So DNA plus these proteins that act as spools to wrap around the DNA is what they call chromatin. Anyone heard of chromatin before? Okay, so chromatin is when the packaging of DNA, the wrapping of DNA around the histones is loosely packaged. Okay, when it's loosely wrapped, it's called what? Chromatin. When the DNA being wrapped around the histone is loosely wrapped around the histone, they call it what? These histones are what? Proteins, all right? Protein spools that the DNA wraps around. So if the DNA is loosely wrapped around it, they call it what? Chromatin. If it's tightly wrapped around it, they call it what? Can someone guess? Anyone heard of chromosome before? So chromosome is what? Tightly what? Packaged. All right. So this is what happens. You have your chromatin all over in the nucleus. Okay. And the DNA, we've already described how it looks like. So structurally, we know how it looks like. Functionally, it contains the genetic information that gives the cell instructions metabolically. It tells the cell what it should do. It tells the cell how it should look like. It gives the cell its instructions to perform its role as a cell. Now, you know that the DNA in all your cells are the same, right? From head to toe. That's why if there's a crime scene and they pick blood and they check the DNA in there, they can take your cheek cells and it should match. right? But your hair cell is producing what? Hair. Your salivary cell is producing what? Saliva. But they both contain what? The same DNA. In other words, the DNA that produces saliva is also in your hair cell. So if your hair cell goes cuckoo, it will start producing what? Saliva too. and your mouth will be producing hairs, right? How are you? And then the hair will be floating out of your mouth, all right? And then you shave it every morning, probably your tongue. But of course, those DNAs have been what? Silenced, all right? So it's only, there are some DNAs that are active and some DNAs that are not active. So your hair cell, the DNA for hair is active and is causing hair to be produced, while your salivary cell, the DNA is what? Silent. So, and of course, there's a lot of detailed mechanics. I teach this pre-med class, and we talk about the details of how that gets regulated. So basically, one of the regulation processes, if so far as one is expressing, automatically the other one gets inhibited. So far as an example I can give you, so far as I contract my biceps, automatically my triceps will relax. Does that make sense? So it's, there are mechanisms like that. That's why two genes cannot be expressed at the same time, because these two muscles cannot relax at the same time. It's something similar to that in ourselves. But anyway, so if DNA is what controls the cell and tells the cell what it should do, it means that somehow that message should get to the cell for the cell to do what it's supposed to do, right? The way that occurs is that the information in the DNA, which we refer to as the genetic codes that have the information, that information must be given over to a messenger to send to the cell. So the information on the DNA, enzymes read it and prepare this other nucleic acid molecule called RNA. And that RNA forms what we call the messenger RNA, because it's the messenger having what? The message that the DNA is trying to give to the cell for the cell to do whatever it's supposed to do. Does that make sense? So inside the nucleus, the DNA gets transcribed into what we call the messenger RNA. And that messenger RNA will come out and enter this membranous stack of things here that have ribosomes attached to them. And they are called rough what? Endoplasmic reticulum. Now, before I go ahead, I want you to understand something. The basic information that this messenger RNA is having is to tell the cell to give the cell a specific instruction to form proteins. So the specific organelle that forms a protein is a ribosome. Ribosomes are the organelles that will synthesize the peptide bonds, will bring about peptide bond formation between the amino acids to form the protein. So if I'm a ribosome, I will read the information on the messenger RNA, and what will I do? I'll put two amino acids together and keep adding amino acids to form the what? Protein based on what I'm reading on the messenger RNA. Are you with me? That process they call translation. Why protein? So let me ask you this. If I should get I should have a meal of, let's say I kill a cow and I eat it. Actually, I like goat, curried goat. No, my favorite is lamb. I can't choose, right? So whatever. I pick an animal, right? I kill it and I eat the whole animal. Let's say chicken, including the feathers, the claws, everything. What source of nutrient would you say I'm having? Well, source of nutrient. Protein. Good. But I ate the whole animal. So, and we know the whole animal is made up of what? Carbohydrates, proteins, lipids, and nucleic acid. But why do you say protein? Because of course, the infrastructure of the whole body or cells are what? Protein. All the others add as what? Modification. Of course, the nucleic acid has its role, but the carbohydrate, lipids all add as modification, and not because they are not primarily probably the most um, obvious thing, but they they bring about the functionality. But the protein, apart from the fact that they have specific functions they play with enzymes, we talked about some of it already, but they also give infrastructure. So let me give you this analogy. Look at this building. We are in which building? B building, right? This is the B building, all right, or whatever, the buildings of hostels. And this classroom is the the building, the blueprint of this classroom was to create PowerPoint presentation. We have tables and chairs. The whole idea, the blueprint was to make this a classroom. But the infrastructure was what putting what these four walls there and a door, and it's made up of what steel and cement. Does that make sense? My office is nothing like this classroom. My office is what has a couch, computer desk, fridge, whatever is in there. It's not a classroom. It has what specific what things. Does that make sense? On it to make it an office. But the infrastructure will still be the same. What would the infrastructure be? Cement and what? Steel and stuff like that. Does that make sense? So these other things that makes this place a classroom is an addition of what modification. Are you seeing the steel and all those things? No. Okay. And whatever additions in my office is making it what? Usable for what it is as an office, making it a function as an office and make this room function as what? A classroom. So that's the same thing. The DNA has the code to create that infrastructure. So basically, when we say gene expression, gene is DNA that gets expressed. So when we say gene expression, it basically means synthesis of protein. That's all it means. Does that make sense? So you see that anytime the DNA code has to do with what? The amino acid it represents. And that amino acid that it represents is connected directly to the messenger RNA that it forms. So you're going to see that there is a genetic code which tends to be universal across all life, where enzymes or ribosomes will read the polynucleotide in sequence of 3. Remember polynucleotide, right? So RNA polynucleotide, it will read it in sequence of 3, and it's reading the nucleotides, paying attention to the bases. So if the first base is A, second base is U, third base is G, it will read it as AUG, and AUG, you look in the genetic code, it represents a specific amino acid. Then it will continue the next, after G, what is there? There's a GGC. Then that GGC, the genetic code will show that it represents another amino acid. Does that make sense? If there is a mutation, and let's say instead of it being GGC, that G fell off, or something happened to it that it couldn't read it, then it will read it as what? GC, and then whatever comes next. Does that make sense? So it's just reading it in a line. Are you getting me? It's in codes of what? 3, 3, 3. And each 3 has an amino acid that represents it. Does that make sense? So the ribosome is what does that. It's a ribosome that will what? Read the messenger RNA, which came from the nucleus when another enzyme read the DNA and formed it. Does that make sense? And the ribosome would read that sequence on the messenger RNA and pick the amino acids and what? Put them together by catalyzing their peptide bond formation. Capiche? Okay. So this thing is taking, so if let's say the messenger RNA, this is the messenger RNA that entered, if this is the messenger RNA that entered the endoplasmic reticulum, that is on the endoplasmic reticulum, the ribosome will attach to it and slide over it. And as it slides over it, it will be reading the sequence and be producing what? Proteins. Does that make sense? The polypeptides. So this polypeptide chain, after it has been formed, okay, so let's say we'll enter the spaces, and we'll travel from one space to another, okay. So this is This is the last bit of the, this is all rough endoplasmic reticulum, all right? This whole part is a rough endoplasmic reticulum. So finally, this protein molecule over here will tear off. This protein molecule is going to tear off. So watch what I'm doing. The membrane will invaginate behind. Do you see what I just did? Okay. And the membrane will close, and this side is going to what? Tear off. And now we are going to have a vesicle, which they call the transporting vesicle, containing what? The protein. Do you all see that? So this is how it's tore off. Let me show it to you over here. So if this is the, okay, if let's say this is the endoplasmic reticulum, and let's say this is the material inside, the protein inside, what is going to happen is that part of the membrane will invaginate like this, okay, invaginate like this and fuse. So this is going to go in, this is going to go in. So basically it's going to look something like this. You see what I just did? Okay. And then this is going to fuse and it's going to tear off, and then we have it like this. Does that make sense? Do you all get that? Okay. So this is what happened right here. And this would be called the transporting vesicle. And it's going to transport this protein to the Golgi body. So this is what? Golgi apparatus, or they call it Golgi body. Same thing. Okay. So what is it going to do? It's going to fuse with the Golgi apparatus right here. It's going to fuse. The part where the transporting vesicle fuses, they call it the forming phase of the part where it fuses on the Golgi body is referred to as the forming phase of the Golgi body. So how does the fusion occur? So let me use this over here. So if, let's say this is the Golgi body, okay, this will travel and fuse. So let's see how the fusion is going to look like. It's going to attach like this. This is the content right in here. These two will fuse together to form just one membrane. So I'm going to use a different color to show how they are forming just one membrane. They fuse and then they'll, so I'm just using one color. So they fuse right here, right? They fuse right here, right? This is where they fuse. And it will tear. So it's going to tear. Do you see that? And then this is going to enter. And now this is going to be part of the membrane. This thing, which was actually green here, right? This was it. It's going to be part of the membrane. Does that make sense? All right. So that's how it's going to enter. And so now the content. So I'm going to use this as the content. So this protein, let's say that's it right now. inside the Golgi apparatus. What is happening in the Golgi apparatus? What's the function of the Golgi apparatus? It's going to alter, modify, add, do whatever it has to do in order to make the protein into its final product. So if this protein is supposed to be hair, it's going to what? Call it, allow it to have sulfide bonds. You get what I'm trying to say? Tertiary, do all those things. If it's supposed to be an enzyme, it's going to add what? Cofactor, add minerals to it, add vitamins to it, whatever it's supposed to add to it to make it whatever it's supposed to become. Does that make sense? If it's supposed to be an antibody, it's going to add carbohydrates to it. Whatever it has to do to modify it for it to become what it's supposed to become. Does that make sense? So the final modification process of that protein to make it into what it was supposed to be gets done where? In the Golgi apparatus, and then it's sort of packages it. So what happens in the Golgi apparatus is that one place, so this thing, when it's taking place inside, part of the membrane will tear off and move and fuse with the next membrane in the middle, and then after the modification to tear off. So for the Golgi apparatus, they keep tearing and joining, moving down this direction. Does that make sense? So it gets to the end, where the end of the beginning is called forming phase. The end of the Golgi apparatus is called maturing phase. And the maturing phase, if this is the final product in this thing right here, if this is the final product, do you see part of the Golgi body is going to tear, and this one will tear off completely and fall out from the Golgi apparatus with the final product inside. And now this will be called a secretory vesicle if it's supposed to secrete the contents out. Does that make sense? Sometimes this is going to form what? An organelle like the lysosome, or it may form the organelle like the peroxisome. Does that make sense? All right. So the rough ER is basically there to house protein, okay, protein formation. The Golgi apparatus is doing what? The modification and packaging of the final product. What about the smooth ER? This is what? Smooth what? Endoplasmic reticulum. The difference is that the smooth has no ribosomes. So guess the biological molecule that it forms. What do you think? If rough ER does the production of protein, what do you think smooth ER will do? Think of the building blocks of the cell. What do you think smooth ER will do? Come on. For DNA and RNA, the nucleus responsibility, right? Nucleic acid. What are the building blocks? Tell me. What are the biological carbohydrates, lipids, proteins, and nucleic acids? So for nucleic acid, it's the business of what? The nucleus, right? What about protein? Rough ER. So what do you think the smooth ER will do? Lipids and carbohydrates. Easy. Okay. So the smooth ER is for what? Anything lipid or carbohydrate, like cholesterol, steroids, just anything lipid or carbohydrate. Does that make sense? It forms that. All right. So lysosomes and peroxisomes. Okay, so smooth endoplasmic reticulum. Is lipids and carbohydrates. Okay, So what's the function of the lysosome? The lysosome contains digestive enzymes, and it helps to break down and destroy infections, pathogens, or anything damaged in the cell. Or when the cell gets infected, it can actually autophagia, destroy the whole cell by releasing its contents into the cell. Okay, while the peroxisome helps to neutralize toxic materials, and it acts as antioxidants as well, contains antioxidant substances, and neutralizes toxic materials that may be inside of the cell. And then the mitochondria. So mitochondria, what do you call mitochondria? The powerhouse of the cell. What does that mean? So you ate your food, right? So let me create some space right here. You ate your food, and your food entered where? Your cell, which contains carbohydrates, lipids, proteins, and what? Nucleic acid, and your vitamins and what? Minerals, which activated your enzymes. They acted as the cofactors and the coenzymes, which made your enzymes active in order to be able to metabolize your food. So you need all these things continually. So this food is floating where? In your cytosol, right? Because when you eat the food, it gets digested, it enters your blood. Where would the blood send the food? Into your cells. So eventually those food is in your cells. So carbohydrates, if you ate mashed potato, polysaccharides, what kind of carbohydrates would you have in your cells? Monosaccharides. Good. If it's proteins, you ate your chicken, steak, whatever. What kind of protein level of complexity are you going to have in your cells? Amino acids. Remember, we looked at all of them, the least level of complexity. What about nucleic acid? You have what? Nucleotides. Does that make sense? All those different basic level of complexities. And lipids, triglycerides. So whatever they are all floating where? In the cytosol of your cell, and your cytosol has all these enzymes. So remember, chemical reaction in the cell is called what? You cannot forget your chemistry. You will need it in all your biology courses. So probably we should call anatomy and physiology bioanatomy. Like we have biochem, right? We should have anatophysiochem. Right? So you cannot forget your chemistry. So anyway, so chemical reaction in the cell is called what? When chemical reaction is taking place in the cell, what do they call it? Metabolism. Good. And we have two types. What are the two types of chemical reactions? Either they are forming or they are breaking. So what are the two types of chemical reaction in our cell? Catabolism or anabolism. So which kind of chemical reaction results in energy release? Catabolism, because you are breaking bonds. Remember, the bonds have the energy. So when you break them, energy gets released. So listen carefully to this. So we have enzymes that will be what? Working on these food. Of course, the first one they will work on for energy is carbohydrates, if carbohydrate is present. So they are breaking the bonds. And what are found in bonds? Energy, and what particle of the atom is involved in bond formation? Each atom has three particles, right? Protons, neutrons, and electrons. Which of them is involved in bond formation? Electrons. Good. So when the enzymes are working on the food, two things will be happening: release of energy And that energy will be trapped in what? ATP, right? Which is the wallet of the cell. If you think of money as energy, ATP is what the wallet. So the energy will be stored in this ATP, in the phosphate bonds of this ATP molecule. And also, the enzymes working on the breaking the bonds will also release what? Electrons. Electrons will be released as well. So remember I told you electrons have the energy, right? So guess what? Let me do this. I'm going to do this in another, let me just do it here. So the enzymes working on the food causing energy to be released and what? Electrons, right? So the energy is stored in what? ATP, and electrons is stored in what we call the electron carrier molecule. So energy is in ATP and electrons are being held by what? Electron carrier molecules. So the electron carrier molecules, we'll look at two of them, NAD and FAD. These are the electron carrier molecules. But remember hydrogen, right? Do you all remember hydrogen? And usually during this whole chemical reaction process, hydrogen loses its electrons and it becomes H plus. Does that make sense? If hydrogen loses its electrons and it forms H plus, that makes sense to you, right? Because anytime you lose an electron, but hydrogen has how many electrons? Just one. So when it loses an electron, it's very desperate because it has no electron whatsoever. So it will chase anything that has electrons. So if it sees an an electron carrier molecule, what do you think? Hydrogen, if I'm hydrogen and I see an electron carrier molecule having electrons, what do you think I want to do? I want to go to that electron carrier molecule. So these electron carrier molecules, when they pick the electrons during chemical reaction or metabolism, they form NADH. This one forms FADH2. For instance, FAD tends to be kind and picks two hydrogen. NAD is like, I'll only pick one hydrogen. So you are going to see that NADH tends to have more energy than FADH2 because two hydrogens are using more of the electrons energy than one hydrogen. So if you look at it. So guess what? All this production of the NAD, FAD thing is taking place in the cytoplasm, but some of it is taking place in the mitochondria. Okay, so guess what happens? All the electron carrier molecules that are produced in the cytoplasm are going to enter the mitochondria. Okay, and again, remember when you see electrons, what are you seeing? Money, energy. Does that make sense? So all these electron carrier molecules will find their way where? Into where? The mitochondria. During the chemical reaction, catabolism, which was breaking down this food molecule, which they call glycolysis and Krebs, which was glycolysis, which was taking place in the cytoplasm, the cytosol. The amount of energy is not that much, which enters, which comes together to form the ATP. It's not that much. But these electron carrier molecules, when they enter the mitochondria, there is a process which involves the hydrogen that was desperately chasing the electrons. That causes an increased release of energy. So much energy that the amount of energy is 95% of what the cytosol can produce. So majority of the energy from one food molecule let's say glucose, 95% comes from the mitochondrial activity, while just 5% of the energy, ATP, comes from the cytoplasm. So therefore, now you understand why they call the mitochondria the powerhouse, the energy house, because these electrons that the carrier molecule took away when the bonds were breaking catabolically enter where the mitochondria, and in there there's another set of activity that takes place that allows a lot of energy to be obtained. Does that make sense? That's a different story that we'll look at later. All right. So that's a function of the mitochondria. So then the last thing in the organelle will be the cytoskeleton. And now everything looks like paint. I don't know why anytime I erase something, it just decides to become thicker. Okay, so the cytoskeleton. We have three cytoskeletons: microfilament, intermediate filament, and microtubules. This is the largest, and this is the smallest. And of course, this is the intermediate. That's what they call intermediate filament in terms of size. And cytoskeleton, they are not made up of bones. The fact that you hear skeleton doesn't mean they are bones or shows tissue. No, they are made up of proteins. And these, like microfilaments, made up of the actin protein and stuff like that. But basically, the cytoskeleton helps to give the cell its structure, and it's also for transport. For instance, remember when the secretive vesicles are formed, they have to fuse with the membrane to come out. A process of endocytosis and exocytosis can occur. So when they fuse with the membrane, before they could fuse with the membrane, the secretive vesicles needed to be transported to the membrane. So what will help them go there is the microtubules. So the microtubules also help with intracellular transportation from one part of the cell to the other. All right. So we are going to visit the slides, and I'm going to just repeat some of the things that, not some, almost all the things that I've said. All right. Yes. It's microfilament, intermediate filament, and microtubules. And you are going to see it again. Okay. So we are just going to run through the slides. If you don't understand anything, you want to stop me, because I've almost mentioned everything. This is just you can do it yourself, but I like doing it. Okay. So cells, the smallest living unit in the body. Okay. The name cell, Robert Hooke was the one who made the rule that everything that is made up of cell is a living thing. So they tend to form the building blocks of living things. We see cells in plants and animals. Cells come from pre-existing cells, of course. One cell divides and it forms what? Two cells. We'll talk about mitosis and cell division. And of course, they need what? The raw materials, which are the building blocks, to be able to give two cells. But at the end of the day, all new cells come from pre-existing cells, because they divide by, the process occurs by cell division, which requires mitosis. So each cell performs its physiological function, and together gives us the function of all the organism as a whole. So this is showing us how they are forming the building blocks, of course, and each cell also has its own building blocks, which we talked about, the molecular building blocks. This is showing us cells dividing to form other cells, so they originate from pre-existing cells. They play vital physiological function individually, and hence, as a collective, they give their whole functionality to the organism. So basically, for us humans, we have the sperm and the egg fusing. The DNA of the sperm joins the DNA of the egg, and then makes it diploid. The cell divide by mitotic division, which mitosis has to do with the DNA. Okay, and then this mitosis followed by cell division, and then we have some of the cells specializing into the 4 different primary tissues that we have. Okay, but all of them came from one cell. So the process by which they form different type of specialized cells is referred to as differentiation. So each of these cells performing their own individual functional, vital, physiological role brings about the collective homeostatic state of the organism. So the specialization of it is referred to as differentiation. So I told you, if you look at a cell, the membrane outside is what? Extracellular, because cells come together to form tissues in between this. The cells are referred to as what? Interstitial fluid, which is part of the extracellular fluid. So the cell component has what? Plasma membrane, cytoplasm. Cytoplasm is the fluidy part and what called cytosol, and the organelle part, which are little structures inside of the cell. So the organelles, we have the ones that have no membranes around them, and the non-membranous ones, so cytoskeletons are example. Microvilli are extensions of the plasma membrane to help increase surface area. So, and of microfilaments sort of helps, few microfilaments are there to help to keep them up there, suspended up there. But microvilli is there to increase surface area. So for instance, this cell could have been like this, right? Occupying this space. But the mere fact that it's been thrown in folds makes it what? Longer. So if I should take from here to stretch, I will only have this. But if it was thrown in folds from here to here and I stretch it, it would be longer. Does that make sense? So it's increasing the surface area. Centrioles are microtubules that are important for cell division. So when a cell is going to divide, right? The 2 cells that form, each of them must have DNA, because every cell needs a DNA to tell it what it should do. Does that make sense? So when a cell is going to divide, that DNA needs to what? Photocopy itself first. The process by which that DNA photocopies itself is called DNA replication. So when it photocopies itself, then now one copy will enter one cell and then the other copy will enter the other cell. So guess what? When the process of replication is taking place, the DNA is loose. It's not tightly packed because the enzymes have to be able to read it correctly, right? So it's in a chromatin state. After the DNA has been duplicated, which they call replication, it's another story altogether in its detail. Guess what happens? Now the DNA is packaged tightly into chromosome. Are you getting this? So that's why all chromosomes, you see them like this, right? X like that, right? Why? Because this is exactly the same as this. So then during mitosis, what happens? They split during anaphase. And this is exactly the same as that for the 6 chromosomes. Does that make sense? All right, and go in there. So what helps with that whole process of dividing and the chromosomes into their respective is essentials. Cilia